All right, so we are officially into chapter 10 in the Pierce text, the sixth edition, and we are slowly starting to learn about the smaller and smaller components associated with genetic material. So we're actually getting down to the basic anatomy in this chapter. So, Chapter 8 was really the beginning of the molecular genetics section where we're looking at the chromosomal mutations and how, if something goes wrong during synapsis in the S phase, that as that DNA molecule progresses through meiosis and mitosis, we can get some really bad things happening that are show up as mutations. So, chapter 10 is taking that concept of chromosomal structures a step further, and we're looking at the chemical nature of the gene. So, we're actually looking at what makes up DNA, RNA, and protein. All right, so the beginning of this chapter in the book actually has a really interesting story about some mummies essentially that they had found in Greenland. And these mummies were remarkably well preserved because Greenland is covered in ice for as part of most of the continent for most of the year. So it really is a very, very cold environment. But regardless of that, for at least 5,000 years, there have been native peoples that have lived there. So one of the tribes was called the Sacac people, and it has been hypothesized that those people actually gave rise to the migration of humans to the Americas. So across the bridges from Greenland into northern Canada and then down into the Americas. So it's really conjecture that at this point that the people in Greenland about 5,000 years ago were the ones that migrated into the U.S. and established our Native American populations. So one interesting thing that scientists were able to find about these people is that they had some of these mummies very, very well preserved, and they were able to actually isolate mitochondrial DNA or not mitochondrial DNA. They were able to isolate hair fragments or hair tufts that still had viable DNA associated with them. And what they ended up finding was that in some of these DNA hair tufts, that when they did genomic analysis on them, that there were males that actually were highly related from a genetic standpoint to people in Siberia. And so that really disregarded the original hypothesis that the Greenland Sacac people actually gave rise to the Native American populations in the Americas. So that was a win for science. So the people that actually that migrated from Greenland didn't migrate into the Americas; they migrated into the Russian continents and created those countries. So the way that they actually did use this DNA was through mitochondria. And so mitochondria are going to be organelles in the cell that actually have their own genome. And we're going to talk a lot about mitochondrial DNA because it's kind of exciting, especially if you're into horse racing. So, but what these scientists did is they used DNA sequences, and they were able to map the DNA, the entire genome of these samples that they found in Greenland, and they related them back to sequences of people that were native to Siberia and those in the northern Alps. And there was a lot of similarity between the test subjects from Greenland and those people from the northern Alps. So it's really quite remarkable that they were even able to do this, because DNA can be degraded really easy if it's not preserved. So the only way it was preserved was by freezing, because Greenland is kind of cold. So they were able to actually extract DNA that was 5,300 years old. So let that sink in a minute. That's pretty amazing science. So they also were able to assess microscopic traces of blood on cloaks, and some other clothing material, as well as some weapons. And they found 4 different strains of human DNA as well. So they also have, been able to hypothesize that there was actually migration taking place and other peoples coming in, and there was very much territorial wars that were going on between these new people and the indigenous people. All right, so switching gears into where we're headed with this is DNA and the chemical nature of DNA. So just keep in mind that there's going to be 4 major characteristics that DNA actually has to do, or that the genetic material actually has to possess in order for it to be successfully transmitted from one generation to another and stabilized. So the first characteristic is that the genetic material must contain complex information. Secondly, that genetic material must replicate faithfully, which means that when it replicates, it does so with very, very few errors. The third characteristic is that the genetic material has to encode for a phenotype. And fourthly, the genetic material must also have the capacity to vary for evolutionary processes and for us to really be able to deal with different pressures that are happening at the environmental level. All right, so one of the earlier studies in the 1940s and 50s was actually conducted by Erwin Charkoff, and he really looked at 5 different organisms and the total amount of adenine, thymine, guanine, and cytosine that was contained in these organisms. So he looked at E. coli cells, yeast cells, sea urchin cells, cells from a rat, and cells from a human. And he assayed, which was very, very basic back in those times, and was able to determine exactly how much adenine an individual organism possessed in E. coli, yeast, sea urchin, rat, and human. And he did that for thymine, guanine, and cytosine. And what he found was that the total bases found in these organisms varied greatly. The one that didn't vary very much was E. coli. It's pretty consistent across the board. Ranges between 23.9, but doesn't go any higher than 26.0. However, as we start looking at yeast and sea urchins and rats and humans and getting into more complex organisms, the amount of adenine, thymine, guanine, and cytosine start to vary. I mean, just look at yeast. So we've got high levels of adenine and thymine and low levels of guanine and cytosine. And then when we jump down to humans, we've got similar amounts of adenine and thymine, but really low amounts of guanine and cytosine. So the conclusions that came from Charkoff's study was that basically, even though there were high variations in the total bases that were in each one of these organisms, the ratio of the bases did not change. So this was one of the first studies that really led to the concept that of DNA or of DNA bases pairing with one another, because the ratios of adenine and thymine were all the same, and the ratios of guanine to cytosine were all the same. So the ratios didn't differ very much at all.